Chapter 14, Section 2, Feeder Pathways for Glycolysis. Now, previous courses, whenever you talk glycolysis, in my mind, they've done you a little bit of disservice because it's always been discussing glucose. And yeah, I've actually had some students who think that when it comes to glycolysis and oxidative, and the whole cellular respiration, that it's only glucose of their diet that gets used there. The amino acids and stuff, that's for other things. And well, no. It's whatever sugars you have taken up can be utilized in glycolysis. All the other biomolecules, the lipids, the amino acids, they can feed into the citric acid cycle. So we're going to look at those. We're going to look here at what can and cannot feed into glycolysis. So when you look at, endogenous molecules, okay, endogenous means it's already within, it's part of the makeup of that organism. Glycogen, starches, we make glycogen, we don't do starches. So starches, that's going to be more plants. Okay, so we're talking about things here, eukaryotic, but not always us, mammals. Okay, because you got to remember glycogen and starches structurally are quite similar. They're just long polymers of glucoses. It's the glycosidic linkages holding them together, and what degree of branching, how are they branching, and things like that. That's going to be the main difference. So you take an enzyme like phosphorylase. It's going to take one of these sugars, release the glucose from these long polymers, and you end up with glucose 1-phosphate. Okay, glucose 1-phosphate is not the first step of glycolysis. Okay, glucose 1-phosphate is close, has all the components, but it needs to be on the 6th place. So you end up with a phosphoglutamase, phosphoglucomutase. Okay, remember mutase just moves stuff around. All the components are still there, they're just which carbon are they on now. Switches. So instead of glucose 1-phosphate, you now have glucose 6-phosphate, and glycolysis of everything we just talked about last section. Then you look at, what you take up in your diet. Different glycogen starches, because you went and you ate a cheeseburger. Okay, cheeseburger with all the fixings. So there's going to be lots of starches from, the lettuce, the tomatoes, the pickles, whatever. There's going to be lots of glycogen because it's a big old hamburger. So start breaking down those, and notice the multiple arrows. So there's multiple steps here in getting things broken down, and you end up with free glucoses. Those glucoses which will immediately feed into glycolysis. Lactose, trehalose, other sugars that may be somewhere in your diet. Lactose gets turned, can be converted into glucose, or lactose can be converted to galactose, which through multiple steps in the liver and using a phosphorylated by UTP, notice UTP, not ATP, you end up with glucose 1-phosphate, which can be converted to glucose 6-phosphate. Maybe you ate a piece of fruit. Fruit's sweet because of high levels of sucrose. Sucrose is a fructose and glucose joined together. Sucrase releases the glucose, boom, goes on, enters into glycolysis. The fructose can then be phosphorylated in the kidneys, muscles, whatever, becomes fructose 6-phosphate. Second step of glycolysis continues on. Or the fructose can, be utilized in the liver, turned into fructose 1-phosphate, glyceraldehyde, or DHAP. So you see different sugars, different things you can eat in different forms in your diet will ultimately feed into glycolysis. All right, so let's look at those individually. Glycogen, okay, it's how animals store sugars. We store them as long polymers. Okay, glycogen phosphorylases, when they say mobilizes glycogen, means it goes through and cuts one glucose at a time, boom, 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 in the process of doing it, you release glucose 1-phosphate. If you happen to be a plant and you're listening to this, good on you. But instead of glycogen, you're making starches. Again, long polymers of glucose, different branching patterns, different bonds allowing for those branching. But you're going to do roughly the same thing. You're going to release sugar by sugar, but instead of releasing glucose 1-phosphate, you're going to be releasing individual glucoses. Glucose 1-phosphate, as I mentioned on the previous slide, is not readily usable by our cells. Glucose 6-phosphate is. So you have to have a mutase. Remember, a mutase is an enzyme that just transfers things, just moves. They still in, the structural formula stays the same, but it's just which carbon. It was on carbon 1, now it's on carbon 2. It's still in the same molecule, it's just where it's at. You end up generating what are referred to as isomers. So you have isomerases. These are going to be a type of mutase that goes and just shifts things around. Okay, not adding, not subtracting, you're just moving things around. We have alpha amylase. Okay, goes and takes the alpha-1,4 glycosidic linkage of starch and glycogen. Okay, glycogen in us. Okay, some starches that are readily available. Okay, you see this breads, pastas, potatoes, certain vegetables, certain plants make this. This alpha-1,4, this is a digestible fiber. We make this. Good yield, maltose, maltose, some dextrins, depending upon which one. Is it alpha-1,4, is it alpha-1,6, things like that. When you go through and you start looking at these hydrolases, they're going to be associated with the membrane lining, the small intestine, okay? Because you need to go through and start taking those long polymers. We can't absorb the long polymers, but if we go cut, we can take up lactose, we can take up sucrose, we can take up dextrin individually, not as a long polymer of thousands or hundreds of thousands that may be present. You got to remember, it was a dehydration reaction to join them all together, and it's a hydrolysis reaction to go and release them one by one. Hydrolysis reactions are driven by hydrolases. So, however many glucoses you're going to want to release from the polymer, that's how many waters you're going to need to have there. Now, cellulase is an enzyme that goes through and breaks the glycosidic linkage known as a beta-1,4. We don't have this, okay? This type of cellulose or starch polymer is what for us would be considered fiber, okay? Indigestible fiber. It's what we take to, honestly, to bulk up our, stool so we can pass, the undigestible, the unwanted remnants, we can pass them out as feces. There are microorganisms, though, that do produce cellulase. For the most part, we don't have these. These you're going to find in ruminant animals, cows, goats, things like that. That they're going to have bacteria and some protists that do this. Another example is termites. Termites eat wood, but the termites themselves don't digest the cellulose in the wood. They have protozoa in their gastrointestinal tract that can. Goes through and cuts sugar by sugar, releasing, logarithmic fold more sugar than those protists can absorb, which is going to be readily available for the ruminant animals or the termites, from my example. Lactase is a funny one. As infants, we humans, we make the enzyme lactase early on because it turns out lactose is one of the primary sugars of mammalian breast milk. I don't know why I said mammalian breast milk because no other animals other than mammals make milk. Anyways, we find this in our milk, mammalian milk. So we have to have an enzyme, lactase, to be able to break it down. One of the things, though, is as we age, we hit a certain point where we begin to lose the ability to express this gene. And, quite a large number of us in the Western world, we seem to like dairy a lot more than the Eastern world. Eastern countries do. We drink a lot, we eat a lot of cheeses and stuff like that. So we have lots of lactose in our diet. We're starting to see, we're studying this phenotype of lactose persistence. These are adults who can, into their 20s, 30s, 60s, whatever, drink lots of milk, have no issues because they're still making enough lactase that it's going to break down, release the lactose. They can, convert the lactose to glucose and they can readily take it up. I'm one of these people who don't. So I'm one of these people who are considered lactose intolerant. I stopped making lactase. So any lactose in my diet, I can't convert. But the bacteria in my large intestine can. And they're going to go through and they're going to do all sorts of nice, weird, funky things. To the degree of people having lactose intolerance, it seems to be this spectrum, low levels, because I guess they make some lactase, so it's not a lot. And other people who make no, and they drink milk or eat certain dairy products, they're basically done for the day. It's one of these things. They now make lactase in pill form, so you can take it, as you eat. It releases the enzyme as the pills dissolve, it gets into your small intestine, breaks it, converts the lactose into glucose, which you can readily take up. There's none left or very little left for the bacteria of your large intestine, no problems. Most people who don't want to take that, who are lactose intolerant, they just cut it out of their diet, which is hard to do because everybody likes a nice slice of cheese or milk or something like that. But as long as they don't intake it, ingest it, no problems the rest of their life. Galactose, it's a product of that lactose hydrolysis. Got to remember, this is very important in the infants, okay? Lactose is going to be in the milk. Galactose plus ATP, stabilized by magnesium, makes galactose 1-phosphate. Galactose 1-phosphate, which is made by the enzyme galactokinase, can then be converted into glucose 1-phosphate, which will then be converted into glucose 6-phosphate and enter into glycolysis. Galactose to glucose 1-phosphate, as I just said, it is a series of reactions, as you see here, from you're using UDP galactose, again, UDP, not an adenosine. Remember I told you last chapter, ATP may be the one that we focus on a lot. Evolutionarily that may be the primary, but it is not the only player. First slide, I showed you UTP is utilized, and here I'm showing it again. UDP is utilized to take galactose and push through the power to make glucose. Difference, galactose and glucose. Well, orientation here at carbon 4. Still a 6-sided ring, so we don't really have to go through and do a whole lot. There's not major structural changes. It's mostly going to be right there. Now, a lot of times students will sit there and say, well, just flip that around. That's all you need to do. And no, because remember, this is above, that's below the plane. And to go and completely switch things around, that's just not something you just readily do. So what you see happens is, the hydroxyl that's up in the alpha, here at carbon 4, this gets removed and becomes a carbonyl. Carbonyl gets replaced because you're oxidizing NAD, you're reducing NAD, boom, taking out those hydrogens, releasing a hydrogen, and the remaining oxygen forms a double bond. You now have a carbonyl, and where you reduced, now you oxidize. You're adding those two hydrogens back, and you now have the beta configuration where the hydroxyl is pointed down or below the plane of the ring. So it's not just, hey, let's just switch it, flip it. Ultimately, it's going to look like you've just flipped it, but you turn into a carbonyl, and then you turn it back into a hydroxyl. Fructose 1-phosphate aldolase, okay? You get fructose, and sometimes it's going to come phosphorylated at the carbon 1. Still can't do anything with it because, remember, it has to be at carbon 6. Fructose 6-phosphate. So the aldolase, is going to at least kick it off. This is going to be something you find in the liver. So you're going to take fructose, you're going to phosphorylate it, but it's in the wrong position. Later enzymes are going to take this and turn it into this fructose 6-phosphate, which can be utilized in glycolysis. Fructose 1-phosphate aldolase takes this, boom, and then breaks it. So you're immediately going from fructose 1-phosphate to dihydroxyacetone phosphate, DHAP, and glyceraldehyde. But the thing is, if you remember, to be utilized in the second phase of glycolysis, this also has to be phosphorylated. So there will be another step involved later where this will have a phosphate group added to it. Fructose 1-phosphate is going to enter in. You're going to have a Triose Kinase. It's going to take that glyceraldehyde. Remember, that was just on the last slide. That isn't there yet. And it's going to phosphorylate. Now you have G3P. So you started off with fructose, and in the liver, phosphorylate it, cut it in half. You're going to have DHAP, which can immediately enter into the glycolysis, the payoff phase. But you also have a glyceraldehyde that's not ready yet. So you use a Triose Kinase, Triose 3-carbon sugar kinase. This one specifically works on glyceraldehyde, turns it into glyceraldehyde 3-phosphate, G3P, which can enter directly into the second phase or the payoff phase of glycolysis. See the same thing, hexokinase takes mannose 6-phosphate, an isomerase converts this into fructose 6-phosphate.